Good evening to everyone gathered here on this international webinar on pioneering contribution on understanding of cancer progression and metastasis. It's me, S. Gauri Lekya from the Department of Biochemistry. On behalf of Mahalashmi Women's College of Arts and Science, I wish a very happy Independence Day to you all. On this very special day, I am welcoming you all for this very interesting webinar session today. We start this session with prayer song.
ஐந்து கரத்தனை ஆனை முகத்தனை இந்தில் இளம் பிறை பாலும் ஏற்றனை நந்தி மகத்தனை ஞான கொழுந்தினை புந்தியில் வைத்து அடி போற்றுகின்றேனே.
நமஸ்தேஸ்து மகாமாயே ஸ்ரீபீఠே சுரபூஜிதே சங்கசக்ர கதாஹஸ்தே மகாலக்ஷ்மி நமோஸ்துதே. நமஸ்தே गरुडारूढे कोलासुरभयङ्करी सर्वपापहरे देवी महालक्ष्मी नमोस्तुते. सर्वज्ञे सर्ववरदे सर्वदुष्टभयङ्करी सर्वदुःखहरे देवी महालक्ष्मी नमोस्तुते. सिद्धिबुद्धिप्रदे देवी मुक्तिमुक्तिप्रदायिनी मन्त्रमूर्ते सदा देवी महालक्ष्मी नमोस्तुते.
Yes, Gauri, can continue. Gauri?
With these all hopes of prayers and blessings, we move on to the session. It's our privilege to have Dr. Lukmanul Hakim Farag sir with us. Now I call upon Ms. Kripavati from the Department of Biochemistry to address the gathering.
Good evening to one and all present here. On behalf of the Biochemistry Department, I welcome our Chairman, Thiru S. Kunjithapadam sir, our Managing Director, Thiru S. K. Thirukumaran sir, our Principal, Dr. N. Bhuma ma'am, and all the faculty members and friends for this session. I take immense pleasure in welcoming our Chief Guest, Dr. Lukmanul Hakim Farag sir, Department of Mathematics and Sciences, College of Arts and Applied Sciences, Dhofar University, Salalah, Oman. Dr. Hakim had worked as an assistant professor in Department of Basic Sciences, College of Applied and Health Sciences in Al Shakia University, and worked as an assistant professor of research in chair of Oman's medicinal plants and marine natural products, Daris Center for Scientific Research and Technology Development in University of Nizwa, Oman. He is honored with many awards and fellowships. He had won National Research Award from Research Council of Oman, awarded as Professional from Developing Country by International Society for Autism Research, Toronto, Canada. Certified as a radiation worker by Nagasaki University, Japan, awarded as a research fellow funded by Research Fellowship in Science for Meritorious Students Program, UGC of India. Dr. Hakim sir is a member of American Chemical Society and active member of American Association of Cancer Research USA. We are really thankful to you, sir, for taking the time out in spite of your busy schedule. I'm sure that we'll feel enriched with our knowledge after completion of this session. Thank you.
Thank you so much. Ma'am, can I start?
Yes, sir, you can start. Yes, you can start.
Yeah. Thank you so much for inviting me for giving lecture. And it's an honor for me to Ms. Bhavya. Thank you so much for inviting me. So basically, I'm a cancer biologist. So I'm working on this field for the past more than 8 years. So here, I'm very delighted and very happy to give a lecture in front of young and enthusiastic students of Mahalashmi College Department of Biochemistry. So let me start my lecture by sharing the screen. Hope you all can see the slides, right? So today's lecture is about, as I told you, so I'm working in the cancer biology field for the past more than 8 years. So I have gone through several reviews and research papers to design my own experiments and to have a new idea how I can work on cancer. So today I'm going to share with you some of the reviews what I have gone through for the past more than 8 years. So let me start. Like what are the topics? So I'm going to cover today. So number one, basics of cancer. So what? And then number two, tumor heterogeneity and microenvironment. Number three, cancer stem cell and its experimental targeting strategy. Number four, pre-metastatic niche formation. Number five, angiogenesis cum metastasis or dissemination. Number five, therapeutic options. And finally, limitations. So let me start first of all, what is cancer? So cancer is nothing but it's a uncontrolled cell proliferation with genomic instability. So basically cancer is what? It's an uncontrolled cell multiplication. So as we know, usually the normal cell undergo mitosis every time whenever the cell become old age or something like that. So when the cell divide, it multiplied many times and then eventually it stops. It happens in the normal cells. But in the case of cancer cells, what happens? So the proliferation or cell division, it won't stop. It doesn't stop. It keep on growing. That's why we say it's a tumor mass. And also they have a very, very genomic instability. Genomic instability means usually under normal circumstances, the normal cell don't undergo a frequent mutations. But in the cancer cells, what happens? They have a lot of mutations in the DNA. So we don't know really what happens during when we get tumor, when people get cancer, we don't know really. So far, there is no answer for that. If you ask me the question like how we know when we can get cancer, what is the probability, what is the possibility? So I don't have answer for that. In some point of time in our life, some of our normal cells inside our body transformed into malignant cell or cancer cell by accumulating so many type of mutations. So that's a big area like cancer genomics or cancer proteomics. So to understand, so from exactly how this normal cell transformed to malignant cell. Slide shows why it is important. Why we have to know about cancer or what? Number one, to protect ourselves by doing self-diagnosis. Number two, to obtain a routine clinical sample such as blood, bone marrow, and other biopsies by convincing patients. Because second point is so important. I'm talking about here about clinical trial, cancer clinical trial. So especially, the country like India, what we have, so we, basically like have a very limited knowledge as far as common people are concerned. So that's why here the enrollment of the clinical trial, the recruitment of patients for clinical trial is very, very in low number. For example, if you take United States or Europe, so there are a lot of people, a lot of cancer patients, they enroll in clinical trial because they know what is cancer and they know what is the value for enrolling them in clinical trial. Number three, to recruit more number of patients, as I told you, because when we don't have enough number of patients, so we cannot test any drug on them, and we cannot come up with some good, concrete conclusion or conclusive evidence. So these are the three important points why we need to know about cancer. this slide was like 1 out of 3 women, 1 out of 3 women and 1 out of 2 men will get diagnosed with cancer with the lifetimes. This is like a like a probability, probability according to the statistics done by American Association of Cancer Research. So this slide shows like how many male and how many female, okay, they diagnosed with a different type of cancer. For example, in the case of male, the prostate cancer is the maximum according to 2015 data, and in the case of female, in the case of female, so breast cancer is a more estimated new cases at 2015, and estimated death also more in both prostate cancer in male and also the breast cancer in female. So again, I'm coming to the point. So cancer develop how and why? This is a very important question. But so far there is no conclusive answer for this, because nobody knows when people get cancer. Suddenly, when we go to the doctor, the clinic, for some other reasons, so they do some X-ray, they do some biopsy test, or they do some blood test. Suddenly the people get cancer. So this is really shock to the people. That's the reason why. I mean, we don't know really when exactly it starts and how it proliferates and how it moves from one place to another place within the body, from one organ to another organ within the body. So far, there is no idea. So this is a big challenge, and we need to do something for this cancer. So now actually, I mean, how they develop. Like, so here in this slide, I can show you how I told you before also. So in some part of our life, some time point of our life, the normal cell become malignant cell by accumulating many different type of mutations. Let us see here, for example. So normal cells here. So when after they undergo different round of mitosis or different round of proliferation. So what they do in each and every round of replication or round of proliferation, they accumulate some amount of mutation. Finally, they accumulate more and more amount of mutation. Finally, what happened because of the mutation in the DNA, the normal cell become malignant cell. So and then this is called tumor cell. So and then after some time, this malignant cell become more vigorous. Acquire more aggressive nature, and then they start to spread to other part of the body. This is what happens. But if you ask me a question, how it happens, how the normal cell becomes suddenly a tumor cell. So what happened in between this area, we don't know really. We don't know really so far. So here again, I wanted to share with you. So like for example, when the embryogenesis starts, the fertilized egg becomes gestation, infancy, childhood, adulthood, and after adulthood, I mean, when we reach the age of 12 or 13-year-old, so what happens? Usually, our somatic cells, they always do the repair and repair the damaging process. So what they do, the somatic cell will divide, multiply, and then they repair the old or damaged cells. This is what happens in our day-to-day life. But in this process, something goes wrong during this proliferation or during this mitosis process. That's the reason why they accumulate more number of mutation in DNA. Finally, they become malignant. They become tumor cells. The problem with this tumor, the problem with this malignant cell, what happens? So there are some therapeutic options are available in terms of chemotherapy, radiotherapy, or surgery. But what happens, especially when we think about the chemotherapy, is the most common therapeutic strategy nowadays, what we have. So what happens, these tumor cells, they respond for the first cycle of treatment. When the people go back to home after treatment and they get recur again. Recur means relapse again, coming back. The cancer will come back again even after the treatment. So this is called recurrence, cancer recurrence. So far, there are so many mechanisms have been, reported by a researcher all over the world globally, but still there is no cure. There is no cure for such type of clinical conditions such as tumor relapse, tumor recurrence, or drug resistance, etc., etc. So next part is metastasis. So metastasis means what? Basically, the definition of metastasis. So the dissemination or spreading of tumor cell from one organ to, or from the host organ to neighbor organ. It is called metastasis. So usually what happens, the tumor cells, they detach from the primary tumor and they mix with the circulation, blood circulation, and they move anywhere in the body. So this is called metastasis. So more than 90% of the death causes by cancer, it is because of metastasis. Usually, so I mean, the tumor can be classified into two types: benign tumor and metastatic tumor. So if any people get benign tumor, it can be easily removed by surgery. It can be easily removed by surgery, or it can be easily removed by chemotherapy. But once the cells start to move from one place to, I mean, the host organ to another part of the body, then it is highly difficult to treat the patients. That's the main reason. Metastasis is the main reason to cause death. So what they do, upon colonization, it interferes with the host physiological function. For example, if anybody has breast cancer, the breast tumor cells, they move to the bone, they move to the lungs, they move to the kidney, they move to the brain. So when these tumor cells move to the different type of organs within the body, what they do, after moving to the particular organ, they start to interfere the physiological function of the organ. Let us say, for example, we are all know, like for example, the function of lungs, it is a respiration. If there is a metastasis in lungs, it means the metastatic cells, they interfere with the lung respiration function. So automatically the patients slowly they lose the respiration capacity. So finally they will completely lose the function of lungs. This is how the metastasis kill the patients. So more than 90% of the death is caused by metastasis. And third point, it pollutes the blood circulatory system. So here in this slide, I can show you, like, there are different type of tumors. I mean, it's reported by MD Anderson Cancer USA. So here you can see a different type of, what are the different type of tumors the human has. Here I'm going to discuss a little bit about hallmarks of cancer. Hallmarks of cancer. It means there are some salient features for cancer cells, tumors. Okay, so these two great professors, Professor Robert Weinberg, MIT from USA, and Professor Douglas from Switzerland, they both define some six different conditions of cancer. Number one, sustaining the proliferative signaling. It means what I have to explain here for you. So what is the meaning of proliferative signaling? So under normal circumstances, what happened? The normal cell divide, multiply, and after several division, it receives a death signal from the extracellular environment, and then they stop division. But in the case of tumor cell, even though they receive death signal from outside, but still they won't respond. They still keep on multiplying. They keep on divide to make a tumor, and evading the suppressors, evading the growth factor suppressor, and also resisting cell death. This is what I told you now. Inducing angiogenesis. Angiogenesis means making a formation of new blood vessel near the tumor, so it can bring all the nutrients, oxygen to the tumor very easily. So this is one of the hallmark of cancer. And also enabling replicative immortality. It means the cancer cell never dies. So usually we know the normal cell, they have a telomeric region in the chromosome. So whenever the cell divide, the
Sir, hello. Sir, we are not able to hear, sir. Hakim, sir.
Yeah,
Sir,
can you,
yes,
yeah,
yes.
yeah,
Yes, now.
yeah, can I start? Can I start?
Yes, sir. You can start.
Yeah. So here's this slide. Okay. So here I just mentioned you about hallmarks of cancer. So can you hear me all? Can you hear me all?
Yes,
Hello?
sir. Yes, sir. We can hear.
Yeah.
But little bit
Yeah.
voice is breaking.
Okay, fine. So here in this slide, so here I'm showing like the experimental results of Robert Weinberg. So in 1981 and 1982, he did some, very excellent experiment. So he took a neuroblastoma cell, that is a neurocancer cell, and he extracted the DNA from that cell and he transferred it into the normal fibroblast. So in this case, what happens? The normal fibroblast become the cancer cells. So what does it mean? The DNA which has mutation. So which has mutation that mutation can make the normal fibroblast into the malignant. In the second experiment, what he did, he treated the normal cell with a carcinogen, and this carcinogen treatment will make the DNA to get multiple mutations in the DNA. So and then they make significant accumulation of mutation. Finally, after this experiment, they found that two important genes. Number one is a RAS oncogene. That gene is responsible to induce tumor in the cancer cells, and also mutation in the tumor suppressor gene, that is P53 gene. So next one about tumor heterogeneity. So tumor heterogeneity means we know heterogeneity means it's a combination of different type of cells. It means tumor is not just a single cell. It's not a single type of cell. It's a multiple type of cell within the tumor. Let us say, for example, cancer cells contain tumor tissue contain not only cancer cell but also dendritic cell, T lymphocytes, macrophages, myofibroblast, cancer stem cell, and mesenchymal stem cells, etc. So it's a group of cell. They become tumor tissue. So each and every cell present within the tumor, they have a specific function to accelerate this tumor progress and dissemination. For example, here in this slide, I can show you here the breast tumor. This is normal breast tissue. Okay, they contain fibroblast. Here, when you compare the normal breast tissue with the ductal in situ adenocarcinoma, here you can see the breast tumor, you have a lot of fibroblast around the breast tumor. So you can see clearly, the tumor microenvironment is different from normal breast tissue. And also here you can see again invasive ductal epithelial carcinoma and advanced invasive carcinoma. Here also you can see a lot of difference between the microenvironment. Here you can see the number of blood capillaries here, and also in advanced invasive ductal carcinoma, it's entirely different. So what I want to say, so the tumors, the cancer cell is just not only doing the job, but also the cells who are associated with the cancer cells, they are playing a very important and key role for the cancer progression and metastasis. This is what we say. It is called tumor microenvironment. Yeah. Again, so what are signaling? Cell signaling is going on between the tumor associated fibroblast and other cells. When you see here, the tumor associated fibroblast release a lot of different type of signaling molecules. For example, TGF means tumor growth factor beta, HGF means hepatocyte growth factor, VGF means vascular endothelial growth factor. Like that, several different type of the signaling molecules secreted by fibroblast, which are tumor associated. And then these signaling molecules will guide the cancer cells to grow more and to metastasis, to spread from one place to another place. And again, here also I can show you. Here you can see this is the pancreatic tumor biopsy histopathology results. You can see here there is a neutrophil inside the pancreatic tumor tissue. So what I want to say, the blood circulating cells such as neutrophil, lymphocytes, T cell, dendritic cells, macrophages, all these cells can infiltrate into the tumor. How? Because when the tumor develops, the primary tumor it's irrespective of tissue origin. Any tumor, when they develop, what they do, slowly they will secrete some chemokines. chemokine. This is, for example, maybe you study chemotaxis in your biochemistry program. So chemotaxis means the migration of cells from one place to another place. So here, what happened? Pancreatic tumor, they secrete some type of chemokines. For example, CCL, CXCL125. So these chemokines will bring the immune cells or blood circulating immune cells into the tumor. But basically we know immune cells, their function of immune cells to attack the antigen. To attack the antigen. When the virus or when bacteria, when they invade inside our body, immediately our immune cells will attack them and neutralize them. But however, here in tumor, what happens? Even though they infiltrate inside our tissue, but still they won't kill the tumor. Instead of killing the tumor, what they do, they are helping the tumor. So this is the big question. This area of research is called tumor immunology. So immune cells, what they do with the tumor? Immune cells, what they do with the tumor? It's a very big question. So each and every immune cells have specific role in the tumor biology. So again, here the neutrophil infiltration. So they again secrete a lot of chemokines and lymphokines, okay, to go inside the tumor and also to help the tumor to progress. And then this slide shows the T cell infiltration, T cell, okay, T helper cell or T regulatory cells. All these cells, they infiltrate into the tumor. But however, instead of killing them, they're helping them some way. So that's why recently they got a Nobel Prize like 2 years ago. Two scientists from one from USA and one from Japan, Professor Honjo from Kyoto University. They got the Nobel Prize to find out the blockade of checkpoints. So what they do, so the T cells they infiltrate the tumor, but however they don't kill the tumor. So there is some communication going between the T cells and tumor cells. Okay. So what they did, the Professor Honjo and another professor from USA, they make an antibody, monoclonal antibody. That antibody block the communication between the T cell and the cancer cell within the tumor, so that the T cell can perform its function. So that what happened, once they block the communication, the T cell will kill the tumor. So in melanoma patients, in skin cancer patients, they have done a lot of clinical trial and they achieved a lot of success. So that's why they got the Nobel Prize. Again, T cell infiltration into the breast tumor. Here you can see this is the immunohistochemistry data. Okay, you can see the arrow mark. Here, these are the T cells within the breast tumor. Again, this slide shows the macrophage infiltration. You can see the brown color spots. All of them are macrophages within the breast tumor. Yeah. Again, this slide shows how the macrophage elicits the signaling response within the tumor once they infiltrate into the tumor. Okay, so here a lot of signaling molecules playing a key role, and they have a key communication between the tumor and the macrophage. Again, this slide shows dendritic cells. Okay, in epithelial ovarian cancer and in melanoma. And again, here the mesenchymal stem cells, they infiltrate into the brain tumor. You can see the immunohistochemistry data here. So this is immunofluorescence staining. Okay, here shows the presence of mesenchymal stem cell inside the brain tumor. Okay. So anyway, so far I just shared with you these immune cells. They enter into the tumor. Instead of killing the tumor, they somehow helping the tumor. Fine. The next one is tumor initiating cells, or you can say cancer stem cells. So tumor initiating cells means, for example, so when as I told you before, we don't know exactly when the tumor starts and when the normal cell become tumor cell, we don't know really. But once the tumor is developed, okay. So in 2011, they found that there are some specific population of cells within the tumor. They are responsible to keep the tumor intact. Intact means, as I told you, we have 3 different therapeutic options: chemotherapy, surgery, and radiation. Okay. So despite of these 3 different therapeutic options, still the cancer will come back because the reason why because of these cancer stem cells. So when we treat the patients with the chemotherapy, when we treat the patients with the radiation or surgery, okay, somehow the clinicians cannot able to target these cancer stem cells within the tumor specifically. So that's why they escape from the treatment and they reside inside the tumor. And after the treatment cycle, after 6 months or 1 year or 2 years or 5 years, we don't know, depending on the tumor type, they come back again. So they are the culprits, real culprits for the relapse, for the tumor relapse, and also for the drug resistance. Yeah, here this slide shows that, okay, some simple, things. As I told you now, stem cells are the real culprit. Cancer stem cells are the real culprit. So why we say it's a cancer stem cells? They are basically cancer cell, but they express some stem cell markers. That's why they called as cancer stem cells. For example, they express OCT4, NANOG, and twist. These are the transcriptional factors which is specifically expressed by stem cells. These cancer cells also expressing these transcriptional factors. That's why we call them as a cancer stem cells. Here you can see in vitro assay, okay, when we culture the cancer stem cells in a growth media, they make a spears. It means they can able to make a completely. One single cancer stem cell is able to make a complete tumor. And if it is there, if the cancer cells don't have this cancer stem cell property, they cannot make whole tumor. Hope you can understand, okay? So that's why the stem cell have the self-renewal capacity. So that's why they can able to recreate the tumor completely. But the normal cancer cell, they cannot do recapitulation of tumor. So this is the major difference between normal cancer cells and cancer stem cells. The same scenario is observed in the in vivo assay also. Yeah, here I can show you some of the cancer stem cell markers. So based on these markers, so we can isolate the different type of cancer stem cells from the patients. For example, for leukemia, CD34, CD38 minus, HLA-DR. So these are the CD. CD means cluster of differentiation. HLA means human leukocyte antigen, as you know. So these are the markers, different markers specifically expressed on cancer stem cells in different tumor types. Here this slide shows one good evidence, okay? It's published in PNAS in 2003. So here what they show, the breast tumor developed from cancer stem cells, but what is the marker? So this cancer cell, the cancer cell which are expressing CD24 minus, the receptor, so they can able to make a tumor very quickly compared to CD24 plus, cluster of differentiation 24 plus. It means that the CD24 plus, it's a normal cancer cell. It don't have any stem cell property. CD24 minus, they are cancer cell, but they have stem cell property. So that's why they can able to make or recapitulate the tumor completely. So that's why they have injected in different type of breast cancer cell in different site in this mice, and they found that CD24 minus can able to make a big tumor mass in the breast area. So this clearly indicates that this cell, CD24 cell, breast cancer stem cell, they have a stem cell property. Okay, how we can target them? In order to eradicate these cancer stem cells. So I would like to share with you, with the young students. So how we can develop a strategy to develop any new drug or to new formulation to target specifically this breast cancer stem cells or any type of cancer stem cells. Number one, so we have to inject the cells. We have to collect the biopsy, human breast tumor biopsy. We have to make single cell suspension, and then we have to inject into the mice, and then they make a tumor. So once the tumor is developed, then we have to breed them. Then we have to breed them to have a different number of mice. And also we need to here observe what we have to do. We have to collect the tumor tissue from mice and prepare single suspension and then inject again. So why I'm doing again here, because we must know whether the tumor what we developed here, they have a recurrence property or not. Okay, this is the reason why we are injecting first here, inducing tumor, and again from this tumor bearing mice we are taking out the biopsy, and then we inject again to see the tumor recurrence property. Okay, so by this way, once we develop the xenograft model or any type of tumor model, then we can test our drug in these mice. And also here you can see this effect of drugs on tumor recurrence, as I told you. Okay, so what we do, we can treat the mice, tumor bearing mice, okay, for a certain period, and then we take the biopsy, make a single suspension, and put them again in another mice. It means if when we take the cells from this tumor, if they develop the tumor in another mice, it means it means the cancer stem cells still alive because of treatment. When we take the cells from here, when we take the cells from treated mice and we inject them again here, if we don't find any tumor in the new mice, it means the cancer stem cells is completely eradicated by our treatment. This is a simple experiment, okay, to understand whether our drug is really targeting the cancer stem cells or not. Again, this one is a xenograph model, okay, by doing FACS, fluorescence activated cell sorting technique. So we can again isolate the breast cancer stem cells after treatment, and then we can do a mammosphere formation assay. Again, stem cell markers to understand whether the cell, what we are targeting with our treatment, so whether we are targeting exactly the breast cancer stem cells or not. So we can perform such type of experiment. So these are the different signaling molecules. So we have to confirm it after treatment. And again, this is the another one. It's called epithelial to mesenchymal transition. It means, so when the cancer cell they detach from the primary tumor, they undergo a lot of different modification, lots of modification. So what they do usually we say carcinoma. Carcinoma means the cancer on epithelial cells. So when the epithelial cells become a tumor cell or carcinoma cells, when they start to move from primary tumor to another organ, what they do, basically they are epithelial cell, but if they want to move, they become mesenchymal cells. This is called epithelial to mesenchymal transition program. This is called EMT. So without this program, no tumor cell can detach from primary tumor. So any targeting strategy, drug targeting strategy can target EMT, epithelial to mesenchymal transition program, can be effective drug formulation to treat the cancer patients. Again, this one shows, okay, different mesenchymal markers and different epithelial markers. So after the treatment of tumor, so we can analyze the marker whether our drug is exactly targeting these markers or not. The next one I would like to tell you about metastasis, okay, metastasis. So metastasis, as I told you, is a dissemination of cells or spreading of cells from primary tumor to another organ. So usually the metastases are two types, micrometastasis and macrometastasis. So basically micrometastasis means when the cells detach from the primary tumor, they mix with the blood circulation, and they reside in the blood vessels for a long time, for 2 years, 3 years or 4 years, we don't know really. So they reside inside the blood vessels for some time, and then they go to the another organ. This type of metastasis is called micrometastasis. Macro means the cells detach from the primary tumor and they reside in the blood vessels for some time, for 3 years, 4 years, and then they make a colony in the organ, in another organ. It is called macrometastasis. So both of them are very dangerous condition, very dangerous clinical condition. So Stephen Packard, okay, so in 1889, so he found there is some seed and soil, hypothesis. He make a seed and soil hypothesis. It's very simple. For example, I told you before also. So breast cancer cells, when they detach from the primary tumor, they go to the lungs, they go to the bone, and they go to the brain. They don't go directly to the colon. They don't go directly to the pancreas. So why they choose the lungs, bone, and brain specifically by breast cancer cells? How? Why? So far there's no idea. So he proposed that maybe the breast cancer cells, when they detach from the primary tumor, they found most suitable environment or ideal environment to grow in another organ, that is bone, lungs, and brain. So still the work is going on to understand why these cells specifically choosing the bone, lungs, and brain for them to make a colony. Here I will show you the steps of metastasis. What happens? Okay, when they detach primary tumor, usually they make angiogenesis. As I told you before, they make a blood vessels and they detach from the primary tumor and they mix with the circulation. They go to the heart for circulation and they go to the lungs. They go to the lungs. Okay, in the lungs, what happens? They adhere with the blood vessels and then they do the extravasation. Finally, they make a colony. So these are the serious steps. It takes a long time. It takes a long time for the tumor cell to make a metastasis. So, but however, we don't know still at which time it happens and how it happens, exactly what is going on. Still, we don't know really. If we know that, we can arrest them. Yeah, again, I want to tell you these are the steps of metastasis. Okay, until they make a colony. So before going to in detail about metastasis, there is called pre-metastatic niche. For example, very simple. When we shift our home from one home to another home, when we shift from one home to another home, before we go to the new home, so we set up everything there. Okay, before we go there and stay there, we make a set up everything. Just like that, the cancer cells that when they detach from the primary tumor, if whether they want to go to the lungs or whether they want to go to the bone or brain, what they do, first of all, they make a hospitality. They make a hospital environment. They make a suitable environment in another organ where they want to go and make a colony. So this is a very complicated process, but somehow it happens inside the body. So they make a suitable environment, ideal environment for them. Then they go there and they make a colony very exceptionally well. So here in this slide, I can show you how the pre-metastatic niche they form. For example, here in the case of tubercular bone, okay, so some tumor cell, when they want to move from primary tumor into the bone, so first what they do, they make all the preparation in the bone, and then these cancer cells they move from the primary tumor and then they come to the bone. So here how they do, there are lots of signaling molecules they involve in this. For example, not only signaling molecules, but also cells. For example, sinusoidal endothelial cells. These are the cells, okay, they're responsible to make a very good environment for the incoming tumor cells, to make them to ask them to come here and stay and make colony. Yeah. And these are the cytoactive molecules, okay? So for example, virulent antigen and growth factors, for example, VEGF, PIGF, etc. All these factors are really responsible to make a pre-metastatic niche formation. Here again, one more example, as I told you before. Here, if any the cells they want to reside or they want to make a colony in the lungs, okay, before they reach the lungs, you can see a lot of cells. For example, EPC, platelets, mesenchymal stem cells, HPC, all these different type of cells, they produce a lot of different kind of cytokines. These cytokines are chemokines. They come early. They come early to the lungs, and they make a good environment for the incoming cells. So this is called pre-metastatic niche formation. Again, one more example, pancreatic cancer. What's happening? How they make pre-metastatic niche in pancreatic cancer? Here there are two types of cell, Kupffer cell and hepatic stellate cell in the pancreatic tumor. They help to make a suitable or environment for the incoming tumor cells. Again, mechanism of tumor cell dissemination. So here, as I told you, so before also I told you, epithelial to mesenchymal transition program. So this program occurs, okay, when the cell want to detach from the primary tumor to the another organs, okay. So in this case, what happens? These cells must undergo this program, and then only they can easily evade from the primary tumor, mix with the circulation, and they go to the distant organs. So this is the immunocytochemistry data published in Cell in 2008, epithelial to mesenchymal transition mechanism. Okay, here E-cadherin, fibronectin, and vimentin. These are the markers. These are the markers for mesenchymal cells. Okay, mesenchymal cells. And for epithelial cell, E-cadherin is epithelial cell. For mesenchymal cells, E-cadherin don't express. In epithelial cells, fibronectin don't express. But in mesenchymal cells, fibronectin and vimentin will express. So this data clearly shows that when cell transform from epithelial to mesenchymal, they lose E-cadherin and acquire the expression of fibronectin and vimentin. And again, this slide also shows the schematic diagram, okay, how the cells are transforming from epithelial cell to mesenchymal cells. And these are the signaling pathways which are responsible to make epithelial to mesenchymal transition program. There are different type of signaling molecules. I don't want to go in detail because we don't have time now. The next is angiogenesis. So angiogenesis means, as I told you, it's a formation of blood vessels. Again, to perform angiogenesis, it's a very complicated process. Okay, so when the tumor forms, obviously they need more oxygen. Obviously they need more nutrients. So in this case, they cannot behave like a normal tissue. They are tumor tissue, so they need more oxygen, more nutrient. In this case, they express more factors which are responsible to make blood vessels. For example, vascular endothelial growth factor, fibroblast growth factor, matrix metalloproteinases. These are the 3 factors responsible to make a blood vessels. So usually they express these 2-3 factors more in order to make more blood vessels. So and also it's a very complicated process. Okay, finally they make more and more number of blood vessels in order to get oxygen and nutrient supply. Again, these are the factors, okay, secreted by cancer stem cell, immune cell, and mesenchymal cells, okay, and all these factors are responsible to make angiogenesis. And as I told you before, cancer stem cells, they are also responsible to make angiogenesis. And therapeutic options, as I told you before, we have 3 different options basically. In addition to that, we have immunotherapy, targeted therapy, hormone therapy, and adaptive T-cell therapy. I will go in quickly. Surgery means, as we know, removal of tumor. Radiation means using gamma radiation or X-ray radiation beam. Chemotherapy, adjuvant. Adjuvant means adjuvant, neoadjuvant, chronomodulation. There are 3 types of chemotherapies are available. Adjuvant means people do the chemotherapy and surgery together. So if they give chemotherapy before surgery, it's called adjuvant chemotherapy. If they give chemotherapy after surgery, it's called neoadjuvant chemotherapy. Chronomodulation means they give a different type of chemotherapy with a sequential order. It's called chronomodulation. So, and apart from this, immunotherapy, targeted therapy, hormone therapy, and adaptive T-cell therapy. So adaptive T-cell therapy means, so what happens, as I told you before, T-cell infiltrate into the tumor, but they don't arrest the cancer cell proliferation. So what they do in this adaptive T-cell therapy, they take a T-cells from the patients, they activate them in the laboratory condition, and then they reinfuse them back into the tumor. So by this way, they activate the T-cells to attack the tumor. This is called adaptive T-cell therapy. So all these different, therapeutic options are available in the clinic right now. So discovery of chemotherapy done by 2 people, that is Dr. Sidney Faber and Dr. Subbarao. Okay, so these are the 2 people actually who started the chemotherapy procedure. So Dr. Faber, he started the procedure with a pediatric tumor, the glioma patients, okay, and Subbarao also started the chemotherapy procedure with the lung tumor patients. And these are the different commercially available chemotherapeutic drugs which we have currently, and unfortunately all of them have side effects. That's the main issue. And immunotherapy, these are the different immunotherapy drugs what we have currently in the clinics, and clinicians, oncologists, they use this different type of immunotherapy drugs to treat the tumors, depending on the tumor condition, tumor type, tumor histopathology. And targeted therapy means using monoclonal antibodies. These are the different antibodies what right now we have in the clinic. So targeted therapy, they work specifically. So depending on the expression of particular tumor cell, using the monoclonal antibody, we can target the particular tumor specifically. By this way, we can avoid that systemic toxicity. Again, hormonal therapy, different hormones therapies are available. Adaptive T-cell therapy, as I told you, collect the T-cells from the patient, okay, activate them in the lab condition, again reinfuse into the patients. So therapeutic limitations, number 1, drug resistance, tumor recurrence, and toxicity. These are the 3 different major complications in cancer drug discovery. So when anyone can overcome these 3 issues, it will be a great thing. So principle of drug resistance, okay. So what happens? Actually, it's a drug efflux. So when we try to treat the tumor by drug, so what happens? The cancer cells having the natural property to efflux are not allowing the drug to reach the inside the cell into the nucleus. Unless until the drug what we treat with the cancer cells, it won't reach the nucleus, it won't damage the DNA. So it cannot kill them completely. This is a major problem in the cancer treatment. So drug efflux mechanism is mainly responsible for the drug resistance. And these are the different cytotoxic agents, and these are the different resistant mechanism. So far people are identified all over the world, and still they are working on it to overcome this. Again, recurrence and relapse. Again, as I told you, there are a lot of signaling molecules that are responsible for the recurrence and relapse. Yeah, finally, I will tell you cancer stem cells. The stem cells are responsible, I told you, cancer stem cells are responsible for the cancer recapitulation even after the treatment. So if we target them, if we eliminate them, so the durable response will be very good. It means like the tumor is free of cancer stem cells, so it won't grow anymore. It won't do recurrence. It won't make new blood vessels, like angiogenesis, and also it will lose the capacity to move from primary tumor, that is metastasis. So cancer stem cells are the real culprits to do all these things. So already there are some available in clinical trial to target cancer stem cells. Okay, BBI 608, BBI 503. These are the drugs are already available commercially to target the cancer stem cells in a particular tumor. These are the common side effects of cancer drugs. Okay, there are different things. And cancer drug discovery scenario. So far, this is what happens: spending millions of dollars, developing a new drug, and contributions from researchers. And finally, these three things: tumor relapse, drug resistance, and toxicity, make the hell situation of researchers. So far we cannot be able to overcome, but still we have a hope. So compared to 50 years before, now we have a lot of advancement in cancer treatment. So nowadays the people, even after diagnosis, they have a very, very long lifespan. They have a good prognosis status. So everything going well. So still the people are working everywhere in the world, and then they work very hard to achieve the goal to overcome these three things: tumor relapse, drug resistance, and toxicity. If any drug having the property of overcoming these three clinical manifestations, it's a good ideal drug for the cancer patients. So the failure happens always. These are the peoples who achieves after massive failures: Edison, Albert Einstein, and Stephen Hawking. Despite of many failures, these people achieve so many things in their life. So we have to learn from them. And also, so I believe personally, the one who sent down the disease, who sent down the remedy. So the Almighty always has a cure for everything, always has a solution for every problem in this world. There is no problem exists in this world without solution. So we people, we humans. So God has given us potential. God has given us a knowledge. So with that potential, with that knowledge, we have to find out the cure. So hopefully we will find the cure for cancer one day. And finally, I would like to end with our great Dr. Abdul Kalam's quotes. All birds find shelter during rain, but eagles avoid the rain by flying above the cloud. Problems are common, but attitude makes difference. So problems are there everywhere. So problems are there in the cancer research. Problems are there in the cancer drug discovery. But still, we have to fight. We have to fight to overcome these issues, to help the cancer patients to come out from their pain, to make them live a happy life. Thank you so much. Hello.
Thank you, sir. Participants, you can post your questions in chat box.
Yes, hello, can you hear me? Hear me, please. Can you hear me? Anyone have questions?
Hello.
Hello, yeah, please tell me.
Good evening, sir. Good evening.
Yeah.
Sir, yours is an excellent and very informative presentation, sir.
Thank you.
I am from Hyderabad, Telangana state.
Okay.
Sir, actually, I am professor in pharmacology in Hyderabad.
Okay,
I
okay.
have one question, sir.
Yeah.
The side effects of cancer treatment is that nausea, vomiting, and hair loss. Is there now, sir?
Yeah. It's a common side effect. It's a very common side effect.
Sir, apart from that one, will there be any severe unbearable pain, sir?
Yeah, that depending on the tumor type and depending on the choice of the treatment. For example, when we go for chemotherapy usually induce side effects like as I told you, nausea, vomiting, etc., etc. But when we go for radiation or surgery, though after post surgical consequences are so many are there. The patients usually have the pain at the surgery site when they go for radiation. So what happened? Their skin become more dry, scaly, and also they have a lot of pain through the route of administration. So things are there. They have a terrible pain. Yes.
No, sir. So one more question, sir. Sir, the oral contraceptives used by women, the oral contraceptives used by women to prevent pregnancy.
Yeah.
Sir, do they cause the cancer, sir? I came to know that they have side effects of causing cancer, sir. So, if at all they cause cancer, what type of cancer they cause, and how to overcome them, sir?
Yeah, basically, contraceptive, yeah, as you told me. So basically, contraceptives, they make a hormonal changes, hormonal imbalance, right, in the body.
Okay, sir. Okay, sir.
That's why they stop ovulation and all these things. But
Okay,
however,
sir.
any chemical which go exogenously from outside to inside our body, they cause some side effects, of course. But contraceptives, really, I don't know exactly, but from my understanding, what I can tell you personally, these contraceptives, yes, if we take for a long time, they may damage significantly. They may damage significantly the uterus cell wall, and also the ability of the ovaries to make ovum. Eventually, they may cause ovarian cancer. Eventually.
Ovarian cancer. Okay, sir.
Yeah.
So how to
Yeah.
overcome them, sir? Then.
Yes, so I mean this is the problem, this is the problem, because we don't know exactly when this tumor develop, when the normal cell become malignant cells. So until it shows a symptom, we never know we have cancer. So what we can do usually, generally I can tell, we have to modify our diet like through natural way. We have to diet. For example, we have to eat a lot of vegetables, fresh vegetables, fruits, antioxidant rich diets. These things will delay the onset of cancer. According to the by many researchers, when we take the food with antioxidant rich foods, so this delay the onset of cancer in our body. So having the healthy lifestyle will always prevent many diseases, not only cancer but also many other different diseases.
Okay, sir. Thank you very much, sir. I am satisfied with your answer, sir. Thank you very much, sir.
Thank you so much. Thank you, brother. That's it.
Any other questions? Okay. Thank you, sir. It's really interesting and informative session today. Now I invite Ms. Abita from the Department of Biochemistry to present vote of thanks.
I extend a very hearty thanks to our chief guest, Dr. Lukmanul Hakim Farooq sir, Assistant Professor, Department of Mathematics and Science, College of Arts and Applied Science, Dhofar University, Oman, who spared time from his busiest schedule to grace this occasion. We had an opportunity to hear your thoughts, and this will surely be going to help us. Your thoughts have enlightened and enhanced our knowledge. I thank our management, our chairman, S. Kunjithapadam sir, our college committee members, Thiru C.V. Harikrishnan sir, our managing directors, Thiru S.K. Thirukumaran sir, and Thiru S.K. Sendil Kumar sir, for permitting us to conduct this international webinar. Special mention to our respective principal, Dr. N. Bhuma ma'am, for being a pillar of strength and a catalyst that stimulates us to do our best. I would also like to thank Dr. Amrin ma'am and Shailaja ma'am, the people who worked behind the screen to make this event happen. With a deep sense of appreciation, we thank the participants of this session. Without them, the session won't be completed. Thank you all.